So hello and welcome to another class of AB Science. This is Abhishek with you. So it is the continuation of the first class of protein synthesis. Today I'll be talking about the initiation of protein synthesis. So this is the second stage as I have described earlier in my classes. So today it is the initiation. Now, as we know that DNA synthesis from 5' to 3', RNA is synthesized from 5' to 3' direction. But in case of protein synthesis, it is amino terminal to carboxyl terminal. So the protein is synthesized from amino terminal to carboxyl terminal. Now, the initiation codon is very essential. That is the starting point present in the mRNA where the first tRNA will bind. Okay, that is called the initiator codon. Now, in case of bacteria, chloroplast and mitochondria, the initiator codon, which is AUG, actually codes for, which is actually coding for methionine but in case of initiation codon, if the AUG is present in the position of initiation codon, that time it will be coding for formyl methionine. So the methionine group is attached with formyl group and then it is termed as a formyl methionine. So if I give you an equation, it will be easy for you, where methionine plus tRNA FMET. Yes, one more thing, the tRNA which is bringing the first amino acid, that is formyl methionine, is very specific as initiator tRNA also. So the initiator tRNA, that is tRNA FMET, is specific to the formyl methionine tRNA or formyl methionine amino acid. Okay, so if I go further, that is methionine tRNA FMET plus AMP plus PPI. So this is the equation, first equation, when the formyl methionine is going to be processed. So here you can see that the methionine group is in the form of methionine, not in the formyl methionine group. So next, one enzyme we require here, that is transformylase, which is the enzyme responsible for transferring the formyl group from N10 formyl tetrahydrofolate. So again, if I write an equation with met tRNA FMET plus N10 formyl tetrahydrofolate, I'm writing the full form, then you will get this formyl group will be transferred to the methionine, that is FMET tRNA FMET plus tetrahydrofolate plus, this step also requires ATP, so AMP plus PPI. Now you see the two steps, in two steps the methionine which is going to be the initiator methionine, that is formyl methionine, has to be modified after the attachment of the particular amino acid to the particular tRNA. So tRNA F-mate, as I have said earlier, is very highly specific to the particular amino acid, that is F-methionine, and the F group is added in the another reaction where the transformylase enzymes is doing the job. So now you have the particular amino acid activated and attached to the particular initiator tRNA. So that you have now. Now what we require next, that is initiation factors, mRNA and ribosomes. So ribosomes are of in case of bacteria, I have already told you, that is bacteria, chloroplast and mitochondria. I am saying about these things. I am not discussing about eukaryotic protein synthesis. So 30S and 50S subunit, initiation factors IF1, 2 and 3, and the mRNA. So the mRNA, which is going to be translated suppose here we have, that is AUG, initiator codon, now the 30th subunit is actually the first unit to bind to the mRNA. Now if I bound the 30th subunit, it requires a particular signal sequence or the specific consensus sequence where it should bind. Because the mRNA, suppose it is of 200 nucleotides or 300 nucleotides, but where it has to bind, that is signified by two mechanisms or the two sequences or the two factors, that is, one is Sine-Dalgarno sequence, and the second one is the 16S ribosomal unit or part of 30S ribosome. So 30S ribosome bears the 16S ribosomal protein, which is actually recognizing the Sine-Dalgarno sequence. Where it is present? It is present in the upstream of the mRNA to be translated from the start codon, actually 8 to 13 nucleotides upstream. Okay. Suppose here it is the consensus sequence present where the 16S ribosomal subunit with the help of 30S will bind. Now from here, if it is bind, three sites we have to know, because 30S and 50S subunit forms three sites within them, that is P, A and E site. So P is for peptide site, E for exit and A for amino acid site. Now I will draw a good picture so that you can understand properly what I am trying to say. So one by one step I will show you. The first step I am going to show you is the 30S ribosomal subunit. Now this is the total portion is 30S ribosomal subunit, which is bound with or which is interfered with IF1 and IF3. These are two initiation factors. I have already told you. Initiation factor 1, 2 and 3 is highly essential for initiating the protein synthesis process. Now IF1 and IF3 is bound to 30S subunit. 30S is bound to the mRNA subunit, mRNA thread, and you can see here this is the P and A site, P for peptidyl and A for amino acid site. I'll write the full form over here, peptidyl site, A site is for amino acid site, and E I have already told you, that is the exit. So this P and A is largely confined or comprised, or the I will say the site which is actually bound by both the tRNA and the actual amino acid which is bound to the tRNA, that is P site. P site is utilized for once only during initiation. Means the first initiator tRNA, that is tRNA F-met, will bind to the P site. Other amino acids or the next amino acids which are going in the line will be binding to the A site. It will never go for the P site. So this is the important thing you should remember, that the P site is for accessing, is for once only. So this P and E site is comprised with the help of 50S and 30S. Means only 30S cannot comprise the site where the initiator codon can bind. So it needs both the subunits to bring the or to bind the initiator codon with the amino acid. Now the E site is largely of, means the E section is present in the 50S ribosomal subunit. So you can see that I am not able to write down or draw the 3D structure of a ribosome. So you should map in your brain or just imagine that this is the 3D structure and how it's going to happen. So first, this is the first thing which is made or which is formed during the protein synthesis. Now I am drawing the second step which is going to take place. So this is the second step of the initiation process, where you can see the new member, that is IF2, with the help of GTP bound with a F-met tRNA. F-met containing the anticodon UAC now binds with the P site. And in this step, IF1 and IF3 will be releasing, and 50S ribosomal subunit is going to bind here. So this total, it is 50S subunit. This is the E site I am drawn here because it is exclusively present in the 50S subunit. Now you can see here that IF1 and IF3 released. What is the function of now IF1, IF3 or IF2? That is, you can understand that IF2 bounds with the GTP. There is an energy molecule which brings the F-met tRNA. F-met, that is the initiator tRNA with the initiating amino acid, now binds with the P in the P site with the AUG codon, and IF3 is now the function of IF3 to resist the 30S and 50S ribosomal combination. So as soon as the 50S ribosomal is now combining, IF3 releases or leaves that site, as well as the IF1 which no longer requires, and so on. After that, after it is bound to the total 50S 30S ribosomal complex, IF2 GTP complex, it is called initiator complex. So the next step, GTP will be hydrolyzed in the process and will be released as GTP plus PI. And also IF2 will be recycled with another amino acid tRNA, or the next amino acid which is going to bind to the A site, not in the P site. And the P site amino acid which is now present in the form will be going to the E site. So these things I will be elaborating in the elongation stage, but up to now this is the initiation process which is going on inside the cell. So if you have any questions, please ask me, and we'll meet you in the next part, that is elongation, and if possible termination also I will include there, or it will take one or more classes more. Thank you.
So if you like my classes, give a big thumbs up and don't forget to subscribe my channel, because these things stimulate me to take futuristic classes for you guys. So thank you and be with me.